The very first thing you should know about biology is that it's the study of life. But honestly, that's not quite the whole story. Life is just a word that we use to describe a bunch of different molecules and chemical reactions that come together in some pretty incredible ways. Now, when we piece all of these molecules together, sugars, fats, proteins, somehow we end up with a living cell. And living cells are some really incredible things. They're organized, they grow, they develop and reproduce, they maintain a constant state of internal conditions, something we call homeostasis. They respond to stimuli, and they contain a heritable genetic code that allows for adaptation and evolution. Now, when you look at a sugar molecule and you ask it, why can't you do any of these things? Life is an emergent property. It's something we never would have predicted when we happened to look at just these individual components. But enough about that. Biology is a type of science, and what we really need to talk about today is the scientific method. Observe, research, hypothesize, test, analyze data, and report conclusions. Rinse and repeat. It's a cycle. There's no end. But really, there's no single correct way of doing science. The scientific method isn't like a meticulous recipe for baking the perfect cake, or is it like a clear set of step-by-step instructions for building a Lego Death Star? The scientific method is actually a lot like the pirate's code, more like guidelines. Well, and it's not quite as simple as that. Sure, any man who falls behind is left behind. We would call that natural selection or survival of the fittest. But it isn't a flexible set of principles developed by some seafaring thugs or Jack Sparrow types. With the scientific method, every step is important. But they're written in a generic enough way that there's many different ways to go through them and to make sure that what you're doing is sound science. Observe, research, hypothesize, test, analyze data, report conclusions. Sure, it might sound vague, but so does live, laugh, love, to be honest. The point is that this stuff is broadly applicable. It's simple, but it's also logical and thorough. When we talk about the scientific method, there are a few key elements beyond that whole observe, research, hypothesize, test, analyze, and report cycle. The first and most important thing is that whatever we're looking at is subject to cause and effect. If I drop a rock, it falls to the earth. If I touch a hot stove, my hand gets burned. If I poke a cat, it hisses at me. This ties in with another theme. The scientific process should be reproducible. It shouldn't matter whether I drop a rock today or tomorrow. It should fall to the earth every time. Now, since we need to be able to test whatever it is we're looking at, and we can test it with an experiment or an observational study, this means that our hypothesis needs to be testable. Like, maybe telepathic dolphins control the weather, or aliens built the pyramids. But we'd never know, because there's no way to test that stuff. A hypothesis also needs to be falsifiable. We should be able to collect data that can prove it wrong. For example, if someone claims that Bill Gates is secretly using the COVID vaccine to implant us with microchips, but these microchips are just too small to detect, then this isn't falsifiable. There's no possible way to collect evidence to reject that claim. Albert Einstein once said, "No amount of experimentation can ever prove me right. A single experiment can prove me wrong." Science is not about getting the right answer. It's about asking the right questions. One other common misconception about science is that it's some sort of belief system. There are no absolute truths in science, only differences in the level of uncertainty, and we'll talk a little bit more about that in a bit. When we apply the scientific method, what we're actually doing is using logic and reasoning to reach conclusions, or make inferences if you're looking for a fancier term. Deductive reasoning applies a general principle to a specific situation. Like, for example, if all mammals have hair, and I have a lizard, well, that lizard must not be a mammal. But that's the great thing about it. These general principles can be tested by observations. The opposite of deductive reasoning is inductive reasoning. What we do here is take specific observations, put them together, and use them to form broader principles. So let's say I go to the animal shelter looking at all the cute puppies that need a home. I notice that terriers, retrievers, Pomeranians, and Shiba Inu all have hair. So I use this information to conclude that all dogs must have hair. This brings us back to a general principle. And remember, general principles are testable. I can go look for more dogs and see if they have hair. Hypothesis-driven science uses deductive reasoning. We keep using the word hypothesis, so I want to make sure everyone knows what it means. A hypothesis is a proposed explanation for a certain set of observations. It fits the facts to the best of our knowledge. Once falsified, or if falsified, we can reject it in favor of some other better hypothesis. But don't forget Einstein's quote, "We never actually prove anything in science. Experiments and observations gather data that may or may not support a hypothesis. If the data support our hypothesis, then we can accept it, for now at least. But if the data don't support our hypothesis, then we might modify or reject it completely." Does this sound kind of repetitive to you? Because it should. The scientific process is iterative, which means we repeat it over and over. Reproducibility, though, is how we can be certain of our results. A hypothesis needs to be able to generate predictions, and it should be testable and falsifiable. Plus, if we do end up falsifying our hypothesis, we should be able to modify it and run it through the scientific method cycle all over again. So, how do you write a hypothesis? The most well-known way of writing one is by using an if-then format. But wait, that can't be right. A hypothesis is a proposed explanation, so we should actually need more detail than that. The correct way to write a hypothesis is based on an if-then statement that's followed by a why statement. Let's do an example together. If plants grow in nitrogen-rich soil, then they will have larger leaves because nitrogen is essential for vegetative growth. Actually, as long as we use the same logic and reasoning, we can write our hypothesis any way we want. We don't even have to use the words if or then. Plants growing in nitrogen-rich soil will have larger leaves because nitrogen is essential for vegetative growth. Let's check to see if this is a valid hypothesis. Can we make predictions? Leaves will be larger for plants grown in nitrogen-rich soils compared to other soils. Is it testable? Definitely. We can easily change the nutrient composition of our soil, grow the plants in that, and then measure leaf size. How about that whole falsifiable thing? Well, absolutely. It's possible for us to reject our hypothesis if we measure the leaves and they ended up being the same size, or maybe they're bigger in the nitrogen-poor soil. And, well, if that were to happen, we could definitely modify our hypothesis later. Or, if our data supported our hypothesis, we might have noticed other changes in plant health or biology along the way. Maybe it turns out that too much nitrogen is actually bad for plants. But we could easily modify our hypothesis if that were the case too. There are two types of hypotheses, null hypotheses and alternative hypotheses. So far, we've been talking about the alternative kind, but a null hypothesis means one of no difference. To return to our plant example, the null hypothesis would say if plants grow in nitrogen-rich soil, then their leaves will be the same size as plants grown in other soils, because nitrogen doesn't affect plant growth. So you can see how null and alternative hypotheses are actually complementary to each other. The null one says nothing will happen, while the alternative one is what you think might actually happen. If you're not really sure what could happen, you might have a whole bunch of alternative hypotheses. After your experiment, you'll be able to reject all of them but one if everything goes according to plan. Usually the goal is to reject a null hypothesis in favor of some alternative, since the purpose of experimentation is to understand the relationships between different variables. We don't always end up rejecting our null hypothesis though, but negative results are still very important in science, even if they don't seem as exciting at first. When we test our hypothesis, we're not actually testing that hypothesis directly. We test the predictions that were associated with it, but semantics. The natural world is affected by many variables, but in an experiment, we only change one variable at a time. The variable that we change is called the independent variable, and in response, we measure a different variable, the one that we think will actually be affected by that change. This response variable is also called a dependent variable because what happens to it depends on whatever change we made before. In our plant experiment, the amount of nitrogen in the soil is the independent variable because that's what we changed. The size of the leaf is the dependent variable because that's the thing that we were measuring. Now, we can divide our plants into two groups. In the control group, we're not going to change the independent variable. We'll leave it at its default setting, so normal levels of nitrogen in the soil. However, in the experimental group, what we're going to do is make that change. In this case, I'm taking out all of the nitrogen that was in the soil. But of course, we can't trust the results of our experiment if we're only looking at these two plants. We should probably have, say, 20 plants in our experimental group and another 20 in our control group. We use the term replication to say how many times we repeat the different conditions in our experiment. Every person is unique, and, well, the same goes for plants. Uncertainty comes from natural variation, and it also comes from exactly how precise we are when we're making our measurements and calculating averages. We need replication so we can isolate the effects of our variable from the randomness and uncertainty of the world around us. Higher replication is almost always better, but there's a limit. It's just not possible to do this experiment with, say, 10,000 plants, unless maybe I had access to an industrial scale greenhouse, I suppose. All right, our experiment helps us test a hypothesis, and a hypothesis generates specific predictions from a more general principle. That was the whole deductive reasoning bit of things that I hope you remember. So, what's the most that we as humans can learn about something? Where do facts come from? In science, a theory is a body of connected concepts, really like complex networks of hypotheses that have been supported time and time again by experimentation and observational science. Theories give us a framework for organizing knowledge and are what scientists accept as fact. To the general public, though, the word theory means something that's a guess. Like, have you ever heard the expression, "it's just a theory"? I have a theory that the moon is made of cheese. But scientists use the term theory completely differently. They take it very, very seriously because it's explaining something. Scientific laws, on the other hand, are a bit different than scientific theories. A law doesn't explain something, it just is, like the law of gravity or the conservation of energy. But I think that's enough for today. We've talked about the scientific method, that whole cycle, remember? Observe, research, hypothesize, test, analyze, and report. We've also talked about deductive reasoning, making predictions, falsifiability, and hypothesis testing. This is core stuff when it comes to understanding and doing biology or any type of science. So hopefully you learned something from all of this and can now get into some different topics like scientific ethics, which is some pretty fascinating stuff. Thanks for watching, and I'll see you next time.